skeletal muscle tissue. That is all about the muscles. Okay, a muscle. Muscle is Latin for little mouse, kind of cute. Muscle is going to be the primary tissue in the heart, in the walls of your hollow organs, things like that. And your skeletal muscle is very, very heavy. It makes up about half of your body's mass. Okay. I'm sure that you guys have heard that muscle weighs more than fat, right? A pound of muscle and a pound of fat are going to look very different. They're still both a pound, but you're going to have a lot more content with a pound of fat. Okay. Muscles have some certain properties that we talk about that make them special. Remember, we talked about things that make epithelial tissue special, things that make connective tissue special. The things that make muscle tissue special is its contractility. Contractility is the ability to contract, to shorten, to get smaller, and excitability. Excitability. Okay, excitability is just the process of being able to be excited by nerve impulses, be able to react to those nerve impulses, depolarize, repolarize, whatever it is that is the intended action. Next thing that muscles are really, really good at is extensibility. Extensibility, that is essentially just having, when you stretch one muscle, the muscle on the opposite side will also stretch. Does a little bit of both. Extensibility is being stretched, and then the last is elasticity. They are good at recoiling, going back to what they were at before they were stretched. Cool. Okay, some terminology that is specific to muscles. The prefix myo or mys, miss, that means muscle. You see myo anywhere, you think muscle. The prefix sarco means flesh. Sarco means flesh. In the muscle tissue, we have the sarcolemma. Lemma means covering, or plasma membrane, sorry. Well, yeah, covering, right? The sarcolemma is going to be equivalent to the plasma membrane of regular cells. Plasma membrane is the same thing as the cell membrane, sorry. And then we have the sarcoplasm. The sarcoplasm is the cytoplasm of muscle cells. The cytoplasm is just that internal jelly substance that all the organelles are hanging out in. Okay. It is the same cytoplasm and plasma membrane that you have in your regular cells, but muscle themed. So they're sarcolemma and sarcoplasm. Cool. Muscle has lots and lots and lots of functions. I hope we remember there are 3 types of muscles: skeletal, smooth, and cardiac. Skeletal muscle its job is to move the body by moving the bones. It's going to be attached to your skeleton. Okay? Smooth muscle is going to squeeze fluid and substances through hollow organs. That's going to be stuff like your esophagus, like your intestines, like your stomach. And that motion that they do is called peristalsis. Peristalsis is the wave-like contraction of hollow organs to move stuff. By stuff, I mean like food, water, poop. Okay. Cool. Another one of the functions of muscles is to open and close body passages. We call those muscles sphincter muscles. You have lots of sphincter muscles all around your body. Your mouth is a sphincter muscle. You have a sphincter muscle right above your stomach. You have a sphincter muscle that controls your poop. You actually have an internal and an external sphincter. Your internal one you do not control. Your external one you do. At least I hope you have learned how to control your external sphincter. Okay? They are going to open to allow passageway of a substance, usually, at least with your mouth, it's food, and will contract to close that pathway. Yeah. A couple of functions of the muscle. They also really, really like to help you maintain your posture and stabilize all your joints. For those of you that were in lab today, part of why my shoulder is so clicky like that is because I have never had to use the muscles of my rotator cuff in order to get my shoulder to work. So I had never really worked those rotator cuff muscles. And because they were so weak, my shoulder said, I don't need to be a shoulder anymore. That'll make sense to my Tuesday crew on Tuesday. Okay? And then last major function of the muscle, it's going to be heat generation. Your skeletal muscle generates a lot of heat. And when you tell someone that you're cold, they'll say, stand up and move around a little bit. That's because moving around a little bit generates heat by your muscles contracting. Yeah? Yeah. This is skeletal muscle tissue. Skeletal muscle. Skeletal muscle tissue gets packaged into skeletal muscles. I'm going to use those terms interchangeably. Skeletal muscle tissue, skeletal muscles, I'm talking about all the same thing. They make up about 40% of your body weight, so it's not an insignificant amount. The cells are striated. That means that they have all those little lines on them. And today you're going to learn what all of those little lines are. The skeletal muscle is innervated by the voluntary division of your nervous system, so it is under your control. I can control, to an extent, how much my muscles move, what they lift, what they do, all that jazz, at least my skeletal muscles. Okay? Which one? That is only located in the walls of the heart? Cardiac. Cardiac muscle tissue. That you will only find in the heart. If you're finding it somewhere else, go to a doctor. Those cells are also going to be striated but their contraction is involuntary. You do not control the contraction of your heart. Thank goodness. Your heart is the only, not the only, one of the few organs that starts working before you are even born and does not stop until you die. Your heart works hard, and if you had to consciously remember to beat your heart every second, I don't know about y'all, but I would be dead. I would forget and be, oh, you get me? So thank goodness it is involuntary. Last type of muscle is going to be smooth muscle tissue. And smooth muscle tissue, again, you're going to find that in the walls of your hollow organs, and it is involuntary, innervated by the involuntary division of your nervous system. Again, if I had to think about digesting my food every time I ate, I would not do it. I would be dead. Okay, so that involuntary stuff is for things that you don't have the time, energy, or bandwidth to think about on a daily basis, that your body, thankfully, thinks about for you. Okay. Each skeletal muscle is an organ of its own. Let me put an asterisk on that. Don't get confused and think that your brachioradialis is a different tissue type, anything like that, than your rectus femoris, something like that. So brachioradialis is in your arm, rectus femoris is in your leg. They're the same tissue type. We just deem them as individual organs because organs are just a bunch of tissues working together. And since muscle is such a composite tissue type, you're going to have epithelial tissue, connective tissue, you're going to have nervous tissue, and you'll have muscle tissue all up in one muscle. They like to call them all separate organs. They're not. They're all your musculoskeletal system. Okay. Your skeletal muscle is also going to have, like I said, connective tissue, blood vessels, and nerves. Okay. Gross anatomy of a skeletal muscle. Okay, you're going to have connective tissues and fascicles. Connective tissues are going to cover and support your muscles, and then those connective tissues will converge and form those kind of fascicles, tendons, ligaments, all of that kind of stuff. All right. We have sheaths of connective tissue that bind the skeletal muscle fibers together. The first layer, the most top layer, is called the epimysium. And that epimysium is made up of dense, regular connective tissue. What do you think the next one's going to be? Just wild guess. Connective tissue sheaths are continuous with tendons. Okay, so all of those sheaths that I was talking about, the epimysium, the paramysium, and the endomysium, they will converge. Let's say they're covering their muscle here. They will converge and make the tendon that attaches it to its bone. This is a bad drawing. Don't at my drawing. Okay. Tendons connect muscle to bone. Ligaments connect bones to bones. That looks even crazier up there than it did on here. So, sorry. Cool? I gotta stop talking and drawing things before I look at the next slide. It's right there for you. And it's much prettier than my version of it. Okay? So you have your muscle fiber. Muscle fiber. Oh, I didn't tell you guys that. Okay. Muscle fiber, you're going to get so many names for muscle fiber, right? But muscle fiber, myocyte, what is the other one? Myofibers. Myofiber, not to be confused with myofibril, different things. These are all going to be names for muscle cells. Also muscle cell. All the same thing. Biologists are not creative until they are. And then they're going to come up with 15 different names for the same one thing. That's the only time biologists want to be creative. Okay? So you can see your muscle fiber, your muscle cell, your myocyte is covered by this sheath. That is your endomysium. That is going to cover every individual muscle cell. The packet of muscle cells that are all kind of grouped together, that is called a fascicle. A fascicle is a bunch of muscle cells grouped together. Okay? And then you have a bunch of fascicles grouped together that make up each individual muscle. And that is covered by the epimysium. Okay? Cool? Muscle fiber is wrapped in endomysium. Fascicle is wrapped in perimysium. Whole muscle is wrapped in epimysium. Nerves and blood vessels. Each skeletal muscle is going to be supplied by branches of one nerve, one artery, and one or more veins. Okay? They come in a bundle. You get a deal. 3 for the price of 1. 3, 2, 7 for the price of 1. Because you're going to have multiple veins. Okay, your nerves and vessels are going to branch repeatedly. And when we get into the nervous system, we're going to talk about how those nerves branch a little bit. Okay? Yeah, I'll talk about that later. Okay? When your muscles attach, your skeletal muscles are going to run from one bone to the other. One bone is going to move, the other bone is going to stay fixed. The one that moves is what we call the insertion. That is the more movable attachment. And then the origin is the less movable. Okay? I'm not going to draw it because your next slide has it. I'm not going to embarrass myself by trying to draw it. I'm not an artist. You don't need to be an artist to be a biologist. Thank goodness. Okay? That is going to be important. For your muscles exam, you will need to be able to tell me the origin, the insertion, and the action of every muscle that we learn. Get ready. I would make some flashcards. Flashcards are going to be your best friend. Origin, insertion, and action. The origin is the part that does not move a lot. The insertion is the part that does move. And the action is what that muscle does. So when you are at the gym, you're working your biceps, you're trying to get swole, trying to get those good, big arms, right? When you are working your bicep, when you lift the weight, you are lifting, you are moving your insertable part. So your bicep inserts right down here in between your radius and your ulna, and it originates way up here at the top of your shoulder. So when I contract, when I want to make this muscle shorter, I am moving my insertion and my origin is staying still. My muscle contracts, I release, my muscle extends back to normal. Okay? So origin and insertion. Oftentimes, your muscle is not directly attached to the bone itself. It's going to be attached via some tendons. And remember, tendons attach muscle to bone. Okay? Well, we have a couple. I'll get there. I'm not getting ahead of myself. We have different types of attachments. There are direct attachments, and that is when the connective tissue fibers are going to be very short. It's going to look like that muscle is directly on the bone, but it's attached by some very short connective tissue fibers. Indirect attachments are going to have some long tendons, or what we call aponeuroses. An aponeuroses is just a sheet of connective tissue. You have a big old aponeuroses that goes from the front of your head to the back of your head. It is called your occipitofrontalis muscle, and it is attached by a very large aponeuroses. Okay. Where bones are pulled on by tendons, that's what's going to make your tubercles, your trochanters, your crests, your tuberosities, your spines, all of that stuff. You get those bumps and I don't know, the only thing that came to my head was bumps and jives, but you get all those bumps and stuff from muscle pulling on that bone. Okay? All right. Microscopic and functional anatomy of the skeletal muscle tissue. Your skeletal muscle fibers, they're going to be long as hell, remember? They're huge cells, and that's why they are multinucleated. Okay. They are formed by embryonic cells fusing together. So you have a ton of those when you were first building your body in utero. All of those embryonic cells, the ones that were said, hey, we're going to become muscles. Great, good job. They all kind of fused together, became one long muscle cell, and then continued doing that over and over and over again to make all of the muscles in your body. These cells are multinucleated, and the nuclei are peripherally located. So they are on the sides, bro. That's supposed to be here, but it's not letting me draw that. You see that. That wasn't my fault. Okay. That's going to be kind of what they look like. So that's going to be your muscle cell that is covered in this sarcolemma. The sarcolemma, again, is just the cell membrane of a muscle cell. Okay. This little guy here, that is called a myofibril. A myofibril. Myofibril. Not a myofiber, that is different. A myofibril. A myofibril is the smallest contractile unit of a muscle cell. It is the smallest piece of a muscle cell that will contract, that will either get longer or get shorter. Myofibril, not to be confused with myofiber. Different things. Myofiber is a bunch of myofibrils locked together in a sarcolemma. Don't worry, I'll say it again. Okay? You get striations. That striation that you see in skeletal muscle and cardiac muscle is from the internal structure of the myofibrils. Okay. Myofibrils, these guys are special structures in muscle cells. I gotta go down a little bit. Took up too much space. They're special structures in muscle cells. You can kind of think of them as an extra organelle that only muscles are going to get. Okay. Long rods, about 80% of your cytoplasm. They're the special contractile organelle found in muscle tissue. A long row of repeating segments of these myofibrils are called sarcomeres. Oh my goodness. Sarcomere. That's not how you spell it. MERES. I'm sorry that sarcomeres is so messy. It didn't want me to write the word. Sarcomeres. You see sarco, you think muscle. Sarcomeres are small segments of contractile muscle tissue. Myofibrils. Okay. Okay. One sarcomere, remember, a myofibril is lots of sarcomeres running together. Okay. One sarcomere, one unit of this contractile space, is going to have what we call a Z disc, either a Z disc or a Z line. You'll hear those interchangeably. They are going to be the boundaries of each sarcomere. Okay. Within that sarcomere, you are going to have actin and myosin filaments. Those are fibers. Those are just types of fibers. Okay. And those fibers are going to allow the muscle to contract. We have this thing that we call the sliding filament theory that I will touch on here, but you'll get a much more in-depth look at the sliding filament theory when you take physiology. Okay. An A band is the full length of the thick filaments and has some of the inner ends of some of the thin filaments. The H zone is going to be the center part of the A band where there are no thin filaments. And then we have this part called the I band. The I band only has thin filaments, and I'm going to talk about it. and then the M line, and I'll touch on those as well. The A bands and the I bands reflect light differently. If you've ever taken a physics class and you looked at polarized light with the thing with the slits, and you turn it one way, you can see it, you turn it the other way, you can't see it, that's pretty much what you're talking about here with those A bands and I bands. If you haven't taken a physics class, just know that it's like looking at something in 3D. Sometimes, so with those 3D glasses, y'all remember those? When they were like red, one eye was red and one eye was blue? It's kind of like that. When you're looking at through the red eye, you can see all of the things that they didn't want you to see with both eyes. When you're looking through just the blue one, you can see all the other stuff. That's kind of that. It's enough of that polarization for you guys to put that together. Okay? Cool. So the M line is the center of the H zone, and then the I band is only thin filaments. I know I shouldn't, but I'm gonna. M line. Oh, that's too long. Those are my thick filaments. And my thin filaments. This way. Notice how my thin filaments are crossing over into the next boundary of this next sarcomere. They're past the Z line, or the Z line. They pop into the next sarcomere. Ooh, that was ugly. Bro. H zone. I think that's everything. Okay. Quick, simple little drawing. Quick. All right. You got your two Z bands that make up the boundaries. You got your thin filaments that get to the other side. Your thick filaments hang out in the middle, and your M line is the middle. The middle line is the middle. There it is. H zone, only thick filaments. A band is whole thick filaments. Z disc is boundary. And this whole thing, I band, only thin. Okay? There it is. That's what it looks like. We also have some extra kind of proteins, molecules, stuff like that, that helps to support your muscles. Titin is one of them. Titin is a spring-like molecule that's going to prevent your muscles from overstretching. Okay. That titin is going to go from the Z disc, one boundary of it, to the M line, to the middle of it. Okay. Holds the thick filaments in place and it unfolds when it is stretched. When it is folded, it's like this. Literally like a slinky. Okay. So that's titin. Your muscles, they have a specialized smooth endoplasmic reticulum. Do you remember what the endoplasmic reticulum does? The endoplasmic reticulum stores stuff and synthesizes stuff. Okay? Your smooth endoplasmic reticulum, that's going to store things like lipids, like maybe not calcium, maybe some like minerals, random things here and there that your cell is going to need to function. In your muscles, we call it the sarcoplasmic reticulum. Okay, that sarcoplasmic reticulum is the specialized smooth endoplasmic reticulum. Okay, it connects with each tubule around each myofibril, the contractile unit, and then those tubules can form things called terminal cisterns. Cisterns. Okay. These cisterns show up in pairs on either side of a T-tubule. And I'll show you what a T-tubule is in a second. Okay. The sarcoplasmic reticulum holds calcium ions. Those calcium ions are going to be released once the muscle is stimulated by a nerve. What will happen is the sarcoplasmic reticulum will release those calcium ions. Those calcium ions, they have a charge to them. Because they have a charge to them, they will depolarize the membrane, and that depolarization of the membrane will force the sliding filament theory to activate and your muscle to contract. Okay? Those calcium ions get absorbed back up by the sarcoplasmic reticulum, and your muscles repolarize and extend again, go back to where they were. T-tubules, those are just deep invaginations of the sarcolemma. Again, the sarcolemma is just the plasma membrane. So that's basically just the plasma membrane said, and did a big dip in and made those T-tubules. Okay. A triad is two T-tubules. Nope, I lied. A triad is a T-tubule flanked by two terminal cisterns. Cistern. And this is a, look more T-like, T-tubule. Okay. There it is. It's not going to let me zoom it in, but remember, the myofibrils are there's a bunch of myofibrils packed together in a sarcolemma to make one singular muscle cell. A bunch of muscle cells packed together makes a fascicle. A bunch of fascicles packed together makes a muscle. Okay. I would take a look at this image a lot closer when you guys have a chance. Okay. Because you can kind of see where the T tubule is. It's right in here. And this is all sarcoplasmic reticulum, and this is sarcoplasmic reticulum. But it's pretty hard to see in this tiny, tiny little guy, but you can blow it up when you're looking at it yourself. Okay? There are two main types of contraction that your muscles do. Concentric contraction, that's going to shorten your muscles to do work. And then eccentric contraction is going to generate, the muscle is going to generate force as it lengthens. Lots of the time, muscles act as a brake. Most of the time, your muscles are actually doing a shortening and pulling rather than a pushing. Okay. Sliding filament theory basically explains, molecular. What am I saying, dude? I need to go to bed. Sliding filament mechanism explains concentric contraction. The myosin head will attach to a thin filament at both ends, pull that filament, and then let it go. The thick and thin filaments do not change size. They stay the same size forever. It is just their location. Okay. Remember, initiated by the release of that calcium ion, and ATP is the thing that makes it happen. Let me try to show you what I mean as best as I can. So let's say I have your arm. Her arm here is my thin filament, okay? My myosin head, once I have been depolarized, my calcium has been released, I am going to grab on to my thin filament. Mind you, my thick filament has not changed length at all. Neither has my thin filament, has not changed length. But now that I have my myosin head, I can pull it this way. When I pull it this way, I am still same length that I was, she's still same length that she was. But because I have pulled this thin filament, my muscle is now shorter, right? This used to be the whole length of it, right? When I attach and I pull, now my muscle is shorter. Does that kind of make sense? That's essentially what the sliding filament theory tells you, or sliding filament mechanism. Once upon a time it was a theory, but I guess they changed the name on me. Happens, okay? That's basically what it is. Myosin head is going to grab that thin filament, pull it, lock it. As long as the muscle is depolarized and still contracting, locked in, eventually that myosin head will release and the thin filament will go back to where it came from. The length of those fibers does not change. It is just their location. They slide over each other. Okay? That contraction changes the striation pattern, right? You see those striations because of how the sarcomeres are built. When you contract your muscles, that pattern changes. There will be no more H zone. The H zone is where there are no thin filaments. That no longer exists. Once your muscle contracts, they're sitting right on top of each other. Okay. Your sarcomere gets shorter, but your A band stays the same. Your A band is the length of your thick filaments. Does not change. Cool? I know we're not cool, but we will be. This is a fully relaxed sarcomere. You have your H band fully intact, no thin filaments hanging out there, living your best life. I band is hanging out kind of this way. Okay. Fully contracted sarcomere, no more H zone. H zone is where there are no thin filaments. That no longer exists here. See that? Okay. Cool. Cool. Muscle extension is where a muscle is stretched by a movement that opposes what it usually does. Okay. Let's go back to the bicep example. Right now, my bicep is contracted. I just want to show off the guns, guys. I've been working out. Anyways, right now my muscle is contracted. If I want to stop contracting my arm, I will have to get this muscle to stretch back out, to extend back out. Once I extend that muscle and that is full length, I'm no longer contracting. There is no more contractile force. This is an extensible force. But this muscle on the back, it is actively contracting when I extend my arm. Right? When you do these guys, your arm is stretched and up, that's when it's heaviest. That's when you're stretching this muscle here. You are creating a force by what to you will look like lengthening the muscle, but in actuality is contracting the muscle. Okay? So that's that. Muscle fiber length and the force of the contraction are all going to be important things to making sure that your muscle is moving adequately. There it is. You got muscle. You got muscle. You got muscle. How are muscles innervated? They are innervated by your nerves, obviously. Thankfully, they're usually the ones that you can control. The spot where the muscle meets the nerve is called the neuromuscular junction. Very creative, right? The neuromuscular junction, where the nerve meets the muscle. The terminal boutons are just the axon terminals. They are going to release their neurotransmitter. And for muscles, that neurotransmitter is always going to be acetylcholine. Acetylcholine. The synaptic cleft is the same thing as the synaptic cleft in your nerves. It is just the space in between the end of the neuron and the beginning of the muscle. And then your neurotransmitters are going to move into the space. Okay? There it is. There it is again. It is a motor unit. We'll talk about motor units a lot when we get to peripheral nervous system in chapter 15, I think.